So again, we don't really know how diseases are caused, but we do know that infectious agents, particles that come in the form of prions, do so because they cause normal proteins in the body to conform to the infectious prions conformation. And so there's a kind of specific prion that is specific to white-tailed deer. And in particular, what happens in deer is that they start to exhibit a lot of weight loss, and they also exhibit abnormalities in terms of their walking patterns, which is very clear and apparent that there's some sort of issue going on. But essentially what you have here is you have a transmissible spongiform encephalopathy that starts to develop in the deer, and then the deer start to waste away, which is why we refer to this as chronic wasting disease. And if you're interested in that, you can look online and kind of observe some of the images that are there. It's kind of pretty horrible kind of infection when you look at it. Okay, so that's our acellular microorganism unit. So now we're going to fall back into the realm of bacteria, which we've talked about before in here, where we have essentially three primary groups: gram-negative bacteria, gram-positive bacteria, and then there's a third group where we have bacteria that do not have a cell wall to them. Remember I talked about this in chapter 3. We have the group known as mycoplasma, which are bacteria that do not contain a cell wall, and because of that we have to use a special staining technique on them called acid-fast stain in order for us to see them and visualize them under the microscope. With bacteria, in terms of how we classify them and understand them, we have to observe cell morphological attributes. We have to observe how we're able to stain different specimens. We observe whether or not bacteria have the ability to be motile, which means they have a flagella and they swim, or they're able to move in some form or fashion, either through a flagella or through tumbling motility, where the cell just looks like it's tumbling like down a hill. Colony morphology, this is observing colonies grown on a petri dish. We also look at the atmospheric requirements that are involved in order to allow bacteria to survive, nutritional requirements, biochemical and metabolic activities, different enzymes being expressed by the bacteria, the pathogenic nature of the bacteria, as well as the genetic makeup of the bacteria. So all of those attributes are what we use to understand further bacteria. And the bacteria of all of these three different organisms, the viruses, the virioids, the prions, and then what we'll talk about next is the archaea group, are the group of organisms that we probably know the most about, because we better understand a lot more attributes of them. So regarding cell morphology, the key characteristic of bacteria is that there are three types of cell shapes primarily. There are the cocci shapes, which are round bacteria, the bacilli-shaped bacteria, which are rod-shaped, which means they're rectangular in nature. And then we also have curved bacteria and spiral-shaped bacteria. And curved bacteria are known as Vibrio bacteria. Spiral-shaped bacteria are known as Beryllium bacteria. Bacteria typically are all unicellular, but in terms of colonization, they will colonize areas with a multitude of cells. So each cell represents one bacteria organism, but all the cells together make up a bacterial species, if that hopefully makes sense to you. So think of it as you have one bacterial cell. That's all you have is a cell. That's still one bacterial species. But most bacteria all are going to be found residing in lots and lots of colonies. And so having that been said, you will see what we call different arrangements, which is how bacterial cells are linked together. You can have arrangement of what's referred to as diplococci, which means you have two coccus cells that are together. You can have streptococci, which would be a chain of bacterial cells all joined together, or a cluster of bacterial cells, which would be called staphylococci. There's also a tetrad structure, and then an octad structure, among other structures that are out there. The majority of these arrangements that you'll see can be used to classify or characterize the name of bacteria. So think of something like Streptococcus pneumonia. If you ever hear that term and you don't know anything about cells and what they look at in a microscope, if you know that strepto means chain and cocci means round, then you know that without even seeing the bacteria in the microscope that you have a round bacterial cell that is arranged in the form of chains. And that's what Streptococci pneumonia would look like under the microscope. Every typical coccus is generally about 1 micrometer in diameter, so that's something that can be expected when you look at bacteria under the microscope. And anytime you have a bacterial cocci, you're typically going to have the word coccus. That's going to be in the scientific name of the organism. And here are some examples of the shapes that we've just been talking about. So here's our cocci bacteria, our bacilli bacteria, and then curve-shaped bacteria. And if you've never seen syphilis under a microscope, this is pretty much what shape you would see if you were looking at the organism that caused syphilis, which is called Treponema pallidum. Bacilli, you just have these rod-shaped. The arrangement here is all singular because you don't see any of the cells linking up together. But here in the coccus shape, you can see clusters of coccus all together, so we refer to this as Staphylococcus. So Staphylococcus aureus, which causes MRSA, and patients would look like this if you were looking at it under the microscope. And here's some more arrangement patterns. Here's the diplococci that I was talking about, some streptococci that are in chains, staphylococci, which are groups, the tetrad, where you have 4 cells joined together, or you can have octads, where you have 8 cells clustered together. And here's some views of actual bacteria that have been viewed underneath the microscope. If you look here, again, at the arrangements of the coccus organisms, you can see some diplococci there. Here's also diplococci. This looks here, you have 4 joined together, so that's a tetrad. All of this would be staphylococci. Not really seeing any strepto, but maybe this these curved cells here. When we talk about a bacterial cell being curved, we're talking about just the cell itself. We're not talking about the arrangement. Okay, so don't get that confused with when you classify or define a colony or arrangement of cells with a cell shape. But you can have a variety of arrangements when you look at a sample of a bacteria underneath the microscope. This is Staphylococcus aureus, so we should see mostly coccus bacteria, which is what we see, and then we also see groups and clusters of bacterial cells joined together. And here's a different microscopic image. This is scanning electron microscopy, and it's taken from an organism called Streptococcus mutans, which is the organism that resides in your oral cavity and is responsible for causing dental caries. And it's existing in the form of different chains that you can see here. And it looks like the chains are overlapping here. So if you look at this very quickly, you would think, oh, well, this is Staphylococcus. But then you give it another glance over and you see mostly that these coccus cells are all existing in chains, hence the name Streptococcus mutans. So lots of stuff that you can see in a microscope. This diagram is just outlining all the different forms that you may be able to see. Again, bacillus-shaped bacteria, strepto, chain arrangement, staphylococcus, coccus bacteria, diplococcus. These are some, looks to be like curved bacteria, so that would be your vibrio. These are kind of a diplococci, but these are larger cells than what you see with the coccus, but still diplo in nature. Here's a filamentous shaped bacteria that you may see. The curving and twisting would be that of characterized by Spirillum. But there is a good variety of various forms of bacteria that you could possibly see underneath the microscope. Okay, so let's talk about some of the bacilli. Bacilli is also known as the rod-shaped bacteria, and they too can exist in diplo formation or strepto formation. So you either have two bacilli cells, or you can have a cluster of bacilli cells. Sometimes you get these very short bacilli cells under the microscope, and sometimes they look coccus to you. So somebody out there in all their brilliant wisdom decided to refer to these very short bacilli as cocobacilli, which means when you look at the microscope, sometimes you can't tell if it's a coccus bacteria or if it's a bacillus bacteria. So if you have to ask that question, then you kind of want to come to the conclusion that cocobacilli bacteria would be the best answer. And there's a number of examples of organisms that reflect this type of cell morphology, such as Escherichia, which is the genre that makes up the E. coli family. That's what the E in E. coli stands for. There's also Klebsiella, which can be a dangerous organism, as well as Proteus organisms. There's Pseudomonas and Haemophilus, as well as Bacillus species that are also fall as good examples for what types of organisms would have a bacilli-shaped type of cell to them if viewed under the microscope. The next group we have is that of the spiral bacteria. As I said before, these can be referred to as spirillum, or spirillum is where you're going to have very apparent twisting cell shape, but you can also have a curved shape, and if you just have a simple curve shaped bacterial cell, then that's going to be known as Vibrio. So think of something like Vibrio cholerae, which causes cholera. That organism cell is going to be a very small curve shape. There's Campylobacteria, as well as Helicobacter bacteria. All three of these subspecies that we have here, these are all genres that make up these different species, but they're all incredibly pathogenic to the human body. You could probably pinpoint that on the shape of the bacteria because of the contortedness of the cells themselves. We also have two other infectious types of genres, the Treponema, which remember I told you causes syphilis, that are going to be spiral shaped, and then you have Borrelia burgdorferi, which is going to cause a disease called Lyme disease, which is brought to animals as well as humans in the form of ticks, because the organism resides in the gut of ticks. And so those are our examples of our spiral bacteria that we have. Here's a kind of great pictogram of what the spiral bacteria would look like underneath the microscope. This is Borrelia hermesii, and this is taken from a blood smear. And this particular organism causes a fever, or relapsing fever, that takes place inside of a host. So you can see the interesting spiral-shaped bacteria. And like I said, we also refer to this as spirillum-shaped. Okay. So a little bit kind of about staining procedures again. We talked about this last class, the capsule stain, the endospore stain, and the flagella stain. Those are all different types of stain techniques that are used to highlight or pinpoint specialized structural components on particular cells. Remember I told you the capsule would consist of a polysaccharide that surrounds the cell, and not all bacteria have this, but some do, so we can use a negative stain to try to capture, or try to observe the capsule. A capsule stain to do that, not a negative stain. You can also use a spore test to test for the presence of endospores, which is important if you want to know whether or not you have this type of bacteria, which might be pretty deadly, floating in the air. And then you can do a flagella stain, which is use a silver stain in order to figure out if the bacteria moves using a flagella. And then there's also a variety of differential staining procedures. The ones we've talked about so far in this class have been gram stain as well as acid-fast stain. And simple stains pretty much just tell us what the shape of the bacteria is, and that uses very simple reagents such as methylene blue or crystal violet to do that. To make a bacterial smear, you have to make sure you fix your bacteria to the slide.